All right, so our next section is spermatogenesis and spermiogenesis. So lecture 5C. So we have to remember there are 3 basic cell types in the testicle. We've already learned about 2 of them and a little bit about the third. So the Leydig cells are going to make testosterone in response to LH. The Sertoli cells are going to nourish and support the sperm in response to FSH. And they're sometimes also called nurse cells or sustentacular cells. So if you see that terminology, it's all talking about the Sertoli cells. And then finally, the spermatogonia. Those are the cells that are going to become sperm. You can think of them as baby sperm. So the spermatogonia are the babies. So let's remember that it's those Leydig cells that are going to make testosterone, and they happen to be located outside of the seminiferous tubules. So what we're looking at right here is a cross-section through a tubule. So this let me use yellow highlighter. So this here is the wall of the tube. We're looking on the inside of the tube, and you can see the Leydig cells are out here. They're just around the outside. That's where they're located in the testes. The spermatogenesis itself, as you know, is going to happen in the seminiferous tubules. And the way it happens is the spermatogonia, the baby sperm, start along the outside walls of the tubes. And then as they undergo meiosis and start to form the sperm, they're going to actually move in toward the middle of the tube, right? So they're going to actually kind of move in toward the middle as they mature and complete that process. So what we're looking at here is, let me use my highlighter again. Right here, I'm going to highlight, whoops, wrong place. Right here is the wall of the tubule. So right up here, oops, that doesn't work. Let me use a different color. So right up here is the lumen, the inside of the tube. So the Sertoli cells, where are they? Well, the Sertoli cells are basically going to, I'm going to use a dark blue pen here, be these weird, I'm going to outline it. It's this cell here, which looks all kind of crazy and weird and branched. So they extend all the way from the bottom, the outside wall of the seminiferous tubule, all the way towards the center of the seminiferous tubule. And those are the cells that are going to nourish and support the sperm and encourage them to do meiosis and to make fully mature sperm. So the Sertoli cells, so you can see that was one. Let me draw another. So the outline of the other one is right here next to it. As you can see, there are these little cells that are in between them that kind of crunch up through, and those little cells are going to be the maturing sperm that are undergoing spermatogenesis. I think it reminds me a lot of like emperor penguins, because like the Sertoli cell is like the adult penguin, and then the little baby spermatogonium is like the little baby penguin. And then here you are, and then as the little baby sperm grows and develops, it's going to move toward the center of the tubule, right? So it gets taller and taller and taller. And although the baby sperm themselves don't get taller and taller, they get closer and closer to the center of the tubule. It just always reminds me of them. So here's another look again at that kind of cross section of the tubule. So right down here, you can see that's the wall of the tubule. And then almost all the yellow that you're seeing with the little speckles in it is the Sertoli cell. So what I'm going to outline for you now is the spermatogonium. So here's a spermatogonium. And what it's going to do is it's going to do duplication and crossover, right? It's going to do the first steps of meiosis. So it does duplication and crossover, and then it becomes, before it does any division, right? So now it becomes a primary spermatocyte. So cyte means cell in biology, so sperm cell. Primary because there's more to come. So that primary spermatocyte then is going to undergo its first division, which biologists call meiosis 1, and form 2 secondary spermatocytes. You can see those 2 secondary spermatocytes. They're going to do the second division, otherwise known as meiosis 2, and form 1, 2, 3, 4 spermatids. Okay, so we start with a spermatogonium, we do duplication and crossover, we become a primary spermatocyte. First division, now we have secondary spermatocytes. Second division, now we have spermatids. So spermatids, as you can see, don't really look like what you think of sperm looking like. So they're right here, and they're these round, fat cells, right? Where are the tails? Where's the sleek, aerodynamic morphology, which is the fancy name for shape? Well, I'll tell you. So anyway, that's where the meiosis happens, and so as it happens, the cells move closer and closer toward the center of the tubule. So in order to slim down and become a sleek swimming machine, those spermatids have to undergo a process called spermiogenesis. How confusing is that, right? So spermatogenesis took us from spermatogonium to the spermatid, and now we do spermiogenesis to get to the full spermatocyte, right? The fully mature sperm. So what's going to happen is that this fat little cell that has all the components of a cell that you ever learned about in biology, so Golgi apparatus and endoplasmic reticulum and lysosomes and all kinds of things, is going to actually get rid of most of its cytoplasm, most of its goo, and most of its organelles, those little cellular parts. Because this guy is just going to turn into a lean, mean chromosome delivering machine. So it's going to keep the nucleus, but it's going to slim it down and make it really compact. And it's going to keep some mitochondria and put those into a mid piece right behind the head. And then it's going to grow a tail. It's going to get rid of all those other cellular pieces of equipment, because you know what? The egg is going to have them, right? So it's like he's moving in with her. He knows she already has the toaster oven, the towels, the sheets, the shower curtain, the dishes. So he doesn't need to bring anything. He's only bringing the DNA. So that's this process of spermiogenesis, is slimming down, growing a tail, getting ready to go and deliver your DNA. The finished product, we have the tail for swimming, right? So it's really cool. This is like the only human cell that actually has a tail and swims. It's pretty awesome to look at. We have that mid piece where we have the mitochondria that make ATP to power the tail. It's going to take a lot of energy to do that. And then in the head of the sperm, we have the nucleus, which has our DNA, our 23 chromosomes, right? So this is a haploid gamete. And then we're also going to have this little acrosome. It's like a little cap, and it's filled with enzymes. And the spermatocyte, this final mature sperm, is going to use the enzymes in that acrosome to burrow into the egg. So that's going to be really important later on. So this is a cool little video of some sperm actually swimming around, so you can look at it on your own time. So in summary, so spermatogenesis, we have a spermatogonium at the outer walls of the seminiferous tubule. It undergoes meiosis, duplication, crossover, first division, second division as it moves toward the center. And it's kind of smushed and held and nourished by those big Sertoli cells that are responding to FSH and stimulating this whole process. And at the end, we get 4 spermatids, right? We get 4 gametes, but they're fat. So then we have to undergo spermiogenesis. They slim down, get rid of their extracellular machinery that they're not going to use. The head is going to keep that nucleus with the 23 chromosomes, the acrosomal cap with enzymes. The midpiece has mitochondria to make energy, and then there's the tail for swimming. So that's it for 5C.